Hi, my name is Steve Sato, University of South Florida in Tampa, and I'm here to talk to you today about the use of silicon carbide for advanced in vivo medical devices. Our research group is involved in a lot of activities, everything from wearables, biosensors, to doing training of the next generation of Bionics biomedical engineering students. Today I want to talk about our neural interface work and also the preliminary work that we have done to show compliance at 7 Tesla MRI. I'm not going to have time, unfortunately, to discuss our nanomedicine work, which is a deep tissue cancer therapy involving silicon carbide nanoparticles. But if anyone's interested in hearing more about that, please contact me and we can discuss it offline. So again, we're going to focus on neural interfaces. And basically, I want to talk about bionics, restoring human functionality. There's a lot of things that we can do to help those who are suffering from limb loss and other types of damage, either from trauma and disease involving the central or peripheral nervous system. Here we see Johnny using an advanced applied physics laboratory MPL robotic arm. Here's Johnny at one of our BME events before the pandemic. Super great guy, amazing individual, has one of the top bionic arms in the world, and we'll talk more about that later. But we're talking about the possibility of using this for prosthetics, using silicon carbide for spinal cord repair, and the so-called neural bridge to basically repair damaged neural circuits in the brain. Johnny will tell you, I'm not going to have time to show the video, that he's able to play the piano, but it's rather slow because of the interface. He's using EMG signals from his upper arm to control the bionic arm and hand. And because of the lack of specificity with the nervous system, he's not able to do truly natural movements. He's very happy with it, but he's not able to do anything even closely resembling his biological arm. So we'll talk more about that in a moment. So looking at some of the old data, this is now about a decade old tissue histology from the brain of an animal. These are computer graphic probes shown where the deep brain stimulation probes were, and we're seeing a lot of damage in this particular animal, both inflammation surrounding where the probe was and a loss of neural tissue where the probe was. So long-term in vivo results that are out there, at least until recently, unless there's something new that I'm not aware of, has shown a big problem with about 50% device failure over time, meaning that half of the implants stop working, sometimes very quickly during the acute phase and oftentimes during the chronic phase. There's tissue damage and there's a loss of functionality and performance. So what we're trying to come up with here is a more robust implant that will perform the same on day 100 that it does on day 1, and ideally would perform for years, especially for human use. That's important. So if we look at what's out there, quite famous and pioneering work done at the University of Michigan in Utah. For silicon, bare-coated silicon devices, they may last 1 to 6 months. Great for neuroscience. This is older technology. And of course, one can use polymer arrays. There's a lot of work going on right now using polymers. They last about a year. But let's face it, polymers are plastics that degrade in harsh environments. The brain's a harsh environment. So one has to question the long-term utility of such devices. But there's always hope, and if that could work, it would be great. But at the moment, it hasn't been demonstrated that there's any long-term functionality. There have been some novel coatings. Amorphous silicon carbide is one of them. They push out the lifetime of the devices to almost half a decade, but clearly for human use, this is insufficient. So we clearly need a new strategy. We believe that cubic silicon carbide, one of the many polymorphs of silicon carbide, may be an excellent choice. We have data that I'll briefly show today that shows excellent neuro and hemocompatibility. And we also have looked at 3 different animal models, mouse, rat, and pig, that show excellent in vivo performance over 1 month. So our in vivo challenge that I've put down in front of my students is the following. We want to demonstrate 25 plus year operation in the human body of silicon carbide implants. And that's when we start to really take advantage of the long-term, robust, corrosion-resistant properties of silicon carbide compared to other methods. So here again is Johnny, and again using EMG sensors to basically pick up signals from the upper arm and then wirelessly send them to both the elbow and the hand. And we believe one solution to this would be such a all silicon carbide, perfectly monolithic silicon carbide implant. Here we see a 16-channel functional implant We've changed our package design. I'll show you that in a moment, but we've done a lot of work with this, including MRI compliance, and I'll talk about that at the end of the talk. So we believe that if we could take this osseointegrated bionic arm and we could literally connect the controls to the nervous system, either peripheral or central with an implant, that we would be able to give Johnny truly biological arm-like functionality that he so deserves, and others who have suffered from limb loss. So our silicon carbide devices, we're right now packaging them for testing, and we'll talk about that later. So this arm is quite interesting. The fact that it's osseointegrated brings a lot of possibilities to the patient, because they're able to perform all of their life functions with this, including ideally taking a shower. Okay. So what about the in vitro performance? We looked at biocompatibility using ISO 10993, and we see that silicon passes the test over 72 hours, or maybe it was 48 hours, but eventually over a longer time, silicon fails the ISO standard. Oxide fails right from the get-go. Cubic silicon carbide and amorphous do quite well. Looking at hemocompatibility, whereas we're looking at platelet attachment and activation on the surface, we see again that oxide is just not a good material to be in contact with the blood. Cubic silicon carbide is excellent, and amorphous is quite good as well. So in the test that we did, this was a DARPA program that we had. We looked at about 7 different materials. I'm not at liberty to disclose what and from whom, but of all the material systems we looked at, only one, cubic silicon carbide, passed all of the tests. And by the way, this ISO 10993 standard is what must be satisfied before going to clinical trial. So it's very important. Both chemical stability as well as bio and hemocompatibility. Only cubic silicon carbide passed the test. So let's talk a little bit about why we think this is useful for biomedical devices. We have 3 times the hardness and 3 times the flexibility of silicon as a basic material, so we can make thinner probes that would produce less surgical trauma. So being able to thin things down and make them smaller scale has been shown to greatly enhance the compatibility of the probe with the brain. Our in vitro data looks great. In fact, our material is as good as Parylene C and polyimide, and we also compared with gold for our conducting heavily doped silicon carbide, and it is as good as standard gold. So we're basically showing in vitro data that is as good as the best materials that are out there. We can also micromachine silicon carbide just as we do silicon. So pretty much any device, topology, architecture that's been realized in silicon, in principle, we can translate that to silicon carbide and get away from the silicon cytotoxicity problem. So here's an example of a 3D array from BlackRock using University of Utah technology. So our goal is to be able to have this type of a device available soon. So what are some of the challenges? And again, going back to this is an old photo from the University Intracortical, University of Utah Intracortical Array. Quite important because it's the only device I believe in the US that's been FDA approved for human trials or human use for those that are chronically or terminally ill. So what happens over time? Well, we can have these platinum coated tips, and there's Parylene C coated shanks. We can have the platinum corrode, exposing cytotoxic or toxic silicon to the brain, which causes failure, or we can have a failure in the coating, which does the same thing. So basically, if we had used silicon carbide, cubic silicon carbide as the support, these types of reliability failures wouldn't really cause us catastrophic failure, and we believe that's why this solution we're proposing makes sense. So again, this cracking and delamination is a challenge. Single unit recordings were not possible with these devices due to reliability challenges. So if we want to have highly accurate neural connection to a device, we need to be able to do single unit recordings, and it's currently not possible with this technology. So why is it that we believe that this would make a great robust microelectrode? Well, again, it's an inert semiconductor, basically a ceramic. We can process it just as NeuroNexus does for their MEAs and implants. We can grow it on silicon wafers, so we can micromachine the same way we do silicon. And here's an example. This is a relatively old picture of some 2 millimeter long blank probes we've done up to 1 centimeter without any problem. So one interesting thing that we can think about is we can use PN junction isolation, where we alternate hole-rich and electron-rich materials. In silicon, we have a very low turn-on voltage of 0.7 of a volt. Great for electronics, not really good for stimulation with a p-n junction diode on a probe. But with silicon carbide, we have a little bit more than double the turn-on voltage. So now you can imagine doing stimulation up to a volt without having to worry about leakage current in the p-n junction. And I'll talk about that a little bit later. So we haven't seen, actually in 3 animal models, this slide has a mistake. We've never seen any chronic inflammatory response. And here's a sample of that. This was 1 month in vivo in a wild-type mouse. And at the site of implantation, it was difficult for Dr. Fruin, who's one of my former students now working at NeuroNexus. He had a difficult time even finding where the hole in the tissue was, where the implant was located. Whereas for the silicon probes, it was rather easy. There was a bright microglial response all around the probe, and there was neural tissue loss for the silicon control, which I'm not showing here. So we're seeing pretty much null response after 30 days, which is quite exciting. I'd like to point out, for those of you not familiar with silicon carbide, it's not just vanilla silicon. There are multiple solid-state forms of silicon carbide. It can be both polycrystalline and amorphous. The amorphous material is what you hear a lot about. We're going to use silicon carbide. Well, they're talking about this amorphous coating. It's an excellent electrical insulator. It can be put down conformally using PECVD typically, and it's been used to enhance the lifetime of many other devices made with polyimide and so on. There's also single crystalline forms, just like you have silicon crystals and wafers. We have this for silicon carbide. There are hexagonal and cubic forms are the most popular, with the cubic silicon carbide form having nearly double the band gap of silicon. So it's quite interesting that we can have access to this material, and it's extremely good in terms of its properties. So we can fashion a device where we basically have the support, the shank, made of single crystalline silicon carbide, so it's able to pierce the brain dura, and it's able to support heavily doped semi-metallic conducting silicon carbide as the conductor to replace the metal, and the amorphous silicon carbide, robust amorphous silicon carbide, to replace the polymers and plastic coatings. So we have a single monolithic system if we do it this way. So that's our goal. One last thing about mechanical properties. Here we see some nano indentation data from the past, where we have basically compared the hardness, the elastic modulus, and the fracture toughness. So we see in all cases that cubic silicon carbide, and these are thin films, compared to bulk material. So it's even more skewed in the advantageous side for silicon carbide. We have about 3 times the hardness, 3 times the elastic modulus, and quite a large factor of improvement over fracture toughness. The material just doesn't like to fracture. And just as a comment, diamond has this problem. Diamond is definitely not fracture tough, and if you start to crack diamond, it will fracture. So one great example of how we can use this, again, we can take a 15 micron thin silicon probe, and we can thin it down to 6 microns and have the same mechanical durability with much thinner cross-section, and therefore much reduced acute damage during surgery. So one bit of data I want to show you is shown here. Here we see a silicon probe glued to a header, surgical header, with the silicon carbide probe glued here, cubic silicon carbide, both the same length, same form factor. After dipping in PBS, due to water tension, this probe literally bent more than 90 degrees to touch the plastic, and after drying, it released. And this was done repeatedly by a colleague, Dr. Joe Pancrazio's student at George Mason, and she kept dipping it into the liquid and kept drying it. And if you see the video, we don't have time for that, it's a rather violent release of the probe, and yet it doesn't shatter. So from a surgical point of view and a mechanical robustness point of view, quite impressive. So let's talk a little bit about how we would replace metals on oxide or on an insulator on silicon with all silicon carbide. So we start with a substrate. In the case of using bulk hexagonal silicon carbide, like 4H silicon carbide, we would take a N or a P-type doped substrate, doesn't matter the polarity. We'd grow a P- low doped hole-rich layer. We'd cap it with a heavily doped N+ layer, which has semi-metallic conduction, and then we cap the whole device using amorphous silicon carbide. Deep reactive ion etching is used to make these mesas. So I'm showing here in this example 3 parallel conductors going into and out of the board. So no plastics, no metals, only robust silicon carbide. So that's the goal. So again, how does this work? I just mentioned the semi-metallic conduction of the heavily doped N plus layer, and then we can think about a lot of devices use, for example, platinum iridium is sputtered or coated on the recording tip to give it ceramic-like properties. Well, cubic silicon carbide is already a ceramic, even though it's a semiconductor. So we don't need to add an extra coating that can over time delaminate and fail. And again, we have this amorphous silicon carbide that's highly insulating. So a test of our system is the following. We're relying on PN junction diodes to prevent leakage currents between those adjacent conductors. And we're also relying on amorphous silicon carbide on the top to cap the device to prevent short circuiting in wet solution like the brain. So here we see an interdigitated electrode or IDE device with two mesas, a P base with an N plus conductor. These are comb drive type structures. So we apply a voltage, measure the leakage current, and we continue to apply the voltage till we get breakdown. And here we see the data for 20 probes. Only 4 of them showed breakdown at less than plus and minus 50 volts, which is quite impressive. And of these that showed breakdown, it was a soft breakdown they recovered, and I believe they all resisted to at least 10 volts, plus or minus 10 volts. And our figure of merit for breakdown was less than a nanoamp of current, which is what we achieved to plus and minus 30 volts, which we thought was pretty impressive. And looking at the histogram, again, we see a majority of our devices, 14 of them didn't leak, even a nanoamp up to plus or minus 50 volts. We had one device that went out to 30, and the rest were basically leaking more current down here. But in every case, it was more than 6 to 10 volts before we measured 1 nanoamp of leakage current. So quite impressive. So how do we make the device? Well, we start out, again, we make our PN junction, we form mesas with deep reactive ion etching, and then we coat with amorphous silicon carbide, followed by opening windows in the amorphous coating with RIE, and that's for the business end, that's for the part that contacts the brain. And for the packaging end, we do use Thai gold to form packaging, a good connection to the package, which would be outside of the brain. So only silicon carbide materials inside the brain. And that's basically our process flow. For the cubic device I'm going to talk about later, it's the identical process flow, only it's a PN junction made on cubic silicon carbide on SOI or bulk silicon. So we used to characterize our devices the following planar MEA. We have single-ended electrodes with a different size of recording tip at the end to look at the geometrical surface area and its effect on performance. And we have double-ended resistors to look at resistivity, and some Schottky and PN diodes to characterize the material. So you'll see a lot of the data you're going to see today is going to be from this structure here. So starting with the 4H silicon carbide, all silicon carbide device, here we see in this blue color the CV curve that we achieved. In magenta is platinum, so we're already exceeding on our first demo device platinum in terms of CV performance, and we're comparing it to Ti nitride and sputtered iridium oxide, which is shown here, so which is quite a good material. And so we perform better than the platinum electrode for 4H. And if we look at the EIS, we're looking at impedance now. I apologize for this busy chart. These are the magnitude of the impedance plots, and this is the phase angle. We're seeing capacitive behavior as we expect for a ceramic-like material, and we're seeing the impedance dropping as a function of the surface area. And at 1 kilohertz, we're talking about 675 kilo ohms for our smallest area device, and we dropped down to about 46 ohms rather for our largest area device. So we're competitive. Even for this non-metal electrode, we're seeing less than a mega ohm for our smallest area device. So we're quite encouraged by that. So we continue our CV story looking at the CV profile as a function of area. So we show that here green is the largest area device, which of course shows the largest hysteresis, if you will, which means charge storage capacity, and blue is hard to read here. It shows our smallest. But what's interesting about silicon carbide, we're doing our standard platinum electrode sweep from minus 0.6 to plus 0.8 volts. If we look at 4H silicon carbide, due to the wider band gap, we can go from minus 2 to plus 2.8 volts easily. So we're able to start to think about stimulating the brain, even up to plus or minus 2 volts. We can think about bimodal stimulation, and so on. So quite interesting possibilities, and please notice the scale difference. So we, of course, as we increase the area, we increase our charge storage capacitance, and we decrease the impedance as expected. So the next step was, we realized that, okay, 4H is working extremely well. I mean, almost no leakage current, wonderful performance, but it's really hard to get through the second hardest material that we know of, which is silicon carbide, to remove that bulk substrate. So one solution to this challenge for manufacturing was, what about cubic silicon carbide and SOI? I've done a lot of work in my group on this over the last 20 years, and then we can use HF to simply release the device. So our films were grown in Catania, Sicily, through a collaboration I've had for nearly 2 decades. And then what this also allows us to do, we could integrate electronics onto the tab outside of the brain, preamplifiers and so on. So this opens up a whole area of technology that we can use because we're using semiconductors for our device. So same idea, we have our PN junction, only now it's on SOI. Here's oxide, there's a thin silicon layer here. And so we make our device, we release with HF. And again, this is the device photo I showed you before. Our next generation, we had a problem with packaging. We've now gone to the same footprint that NeuroNexus uses, and we're partnering with them to package our devices. So here we see the pre-probe definition, etch, and release state. These devices are actually being run through our DRI tool this week. And once we do that, we'll flip, thin the silicon off the substrate, and release with HF. So we're looking forward to seeing how these implants work. And that should be reported at the next conference, hopefully. So what do we have so far for data? Using the same planar MEA, we see that we have nice impedance performance. We have a lower impedance for the smallest area. Before it was in hundreds of kilo ohms, now we're about 165. And then for our CV data, we're showing also nice performance. And if we look at the charge storage capacity for both the anodic and cathodic phases, we're seeing very nice results, which, by the way, are on par with titanium nitride. So now our next generation of all silicon carbide monolithic probe devices seems to be performing as well in preliminary data as ti nitride, which is quite exciting. So the last topic I want to discuss, we're running out of time, is the issue of MRI compatibility. Here we see some MRI data showing damage caused by implants in the brain. In 2012, while I was on a Science Without Borders project in Brazil for this topic, we put some devices in rat brain at 2 Tesla, and we saw a large amount of image artifacts from the so-called platinum iridium MRI compatible device, which you can see here they've twisted the wire to try to reduce the coupling, whereas our bare silicon carbide showed no artifacts whatsoever. So this gave us the idea to follow this further, and what we've done is we've looked at we now have done the experiments and done a lot of simulations, and here we see our implant. In this case, there was a 27 micron thin device layer from the SOI on the backside of the implant. When we looked at both sagittal and coronal views in 7 Tesla MRI, we did actually see a little bit of artifacts at the tip, which was caused by the silicon on the backside. When we removed the silicon, now we have just silicon carbide. Now we see barely any evidence at all that the probe's even in the tissue phantom. This is a gel tissue phantom with electrical properties to mimic that of the brain: permittivity, conductivity, and magnetic permeability. So we just published this results in Micro Machines, and I encourage you to take a look at this because we're quite excited about this. So in summary, we have silicon carbide, which is highly compatible with the brain. We believe this from our in vitro and in vivo studies where we saw no immune system trigger occurring. It's possible to create a monolithic I&I with only silicon carbide, no plastics or polymers, no metals. We use PN junctions to isolate traces through the substrate, and amorphous as our capping layer. And again, our IDE data showed outstanding performance with leakage current less than a nanoamp, out to plus or minus 50 volts for many of our devices. Was quite impressive. We've now translated that hexagonal silicon carbide work to cubic, as shown here. We've been able to easily harvest our device using an HF dip. We had higher charge storage capacity than 4H, but we did have higher leakage current in our first generation cubic devices. Now we're working on Gen 2. I showed you the image before, and it's awaiting final DRI probe definition, etching, and packaging and testing. And our preliminary MRI compatibility data is extremely encouraging at 7 Tesla. No image artifacts. What I didn't show due to time is we did numerical simulations that showed that we're below the specific absorption rate limit by the FDA. We're below that limit for cubic silicon carbide. Other materials such as titanium and so on are not. And our in vivo confirmation of this is planned for this summer if everything works out well with the pandemic. So I have a lot of people to thank. In blue, I'm going to stop here. I'm going to thank all of my colleagues and former students who are still collaborating on this project. And our financial support comes from a number of sources. And here's our group from spring of 2020, already socially distancing with individual photos. And number 16, Mohammed Beji, he's the man behind the MRI work. He did an outstanding job, and we're still collaborating while he's working in the French part of Switzerland, in Lucerne. And I'd like to thank everyone for your attention, and I'd be happy to take any questions you might have.